This is a video recording for Purdue University for Animal Science 345. Today I'm going to be talking about bacteriophage and their use in the animal industry as an alternative to antibiotics. So to begin, I'm just going to talk about what a bacteriophage really is. They are the most common biological agent on Earth. They are found in absolutely every corner of the planet, on every continent, in the oceans, all the way into the crust of the Earth. They are everywhere. And they're a biological agent and not an organism because they cannot replicate themselves. They're actually a kind of virus or an obligate parasite that only infects and replicates within bacterial cells. So they do not harm animals or humans. However, when I say they're a particular virus, it's actually very broad and a general understanding because there are actually hundreds or even thousands of different sizes, different morphologies, and genetic organizations of these creatures. So after knowing what bacteriophage are, here's what they look like. This is going to be the general structure of a bacteriophage. It will include a capsid head full of nucleic acid that is attached by the collar to the sheath, and then to the base plate, tail fiber, and spikes. The tail fibers are going to be used to grab onto bacteria, and the spikes are going to be used to insert into the bacteria. This is basically a living syringe. That's how the body functions when it attaches to a bacteria. So now, after knowing what a bacteriophage is, it's also important to go into the evolutionary and the genetic history of these biological agents. So as I've said before, these are actually kind of a virus, a particular kind of virus, that can only infect and replicate within bacterial cells. This has been going on for billions, if not trillions of years, and it's actually been termed the world's longest arms race between a bacteria and a bacteriophage. So how this breaks down is a bacteriophage or a phage will find a host, and then it'll insert its nucleic acid, either DNA or RNA, and it'll go into a lytic or lysogenic cycle. So in a lytic cycle, the DNA or RNA will immediately go to work on the ribosomes and the other organelles within the bacteria and take it over and use the bacteria as a factory to create its own proteins. Then once it's done, it'll cause the cell to burst and release the new phage to find a new host. In the lysogenic cycle, the DNA or RNA will actually be worked on and then move into the genome of the bacteria. And then after a while, it'll still make the proteins that it's needed, but it won't kill the bacteria right off the bat. It'll actually use it until there's some kind of change, and then it'll move into the lytic cycle and burst the cell. But bacteria have made defenses over the years, so they have inhibition, so they can inhibit attachment, or they can inhibit at assembly. So they have certain proteins and other things on their surface to try and get it to stop, and then they can also interfere with enzymes to stop the proteins from being made. Besides that, cells can also have a cascade effect and cause their cells to lyse at the presence of DNA of bacteriophage. And then they can also have this thing called an RM system, or a restriction modification system, where they can, once the DNA or RNA is entered, they can make modifications or restrictions to what can actually be translated and transcribed to make proteins. So the DNA will go in, but it won't do anything. And then this has just continued over the years. So it's attack, defend, evolve, repeat. That's what this world's longest arms race is. Finding new ways to attack and then new ways to defend and continue on. So knowing about the world's longest arms race, this can actually help us in regular medicine and veterinary medicine, because currently we have an issue, which is antibiotic resistance. So a lot of our infections we have now, over many years we've thrown antibiotics at it, and it's worked, but through evolution, a lot of the bacteria that's caused these infections is actually becoming resistant. There's some that are resistant to multiple or just one type of antibiotic, but it's still bad and it's not helping. So like in the animal industry, in veterinary medicine, some of the bacteria that actually have resistance already are Campylobacter, Salmonella, and the bacteria that causes mastitis. They have some resistance to antibiotics, so this can cause problems for our animals, and also economically, if we can't make milk, it's a problem. So the best way we can use bacteriophage is to eliminate bacteria. So we can actually make a cocktail of phage and insert it and inject it into an area to try and kill the bacteria. But if the bacteria are able to fight back, then we can also use antibiotics hand in hand with that, because if a bacteria is fighting a phage, it cannot at the same time fight against antibiotics. So this is a new way we can go to try and go about killing bacteria and getting rid of infections. This will improve food security and help the world at large. But we can also use DNA changes. So bacteriophage are a way that we can insert new good DNA into some healthy bacteria possibly, and then make the changes from within. So through use of experimental measures and phage therapy, it's already been seen that bacteriophage are a great antibiotic alternative. However, there are some major hurdles to jump through, and the biggest one is legislation. There's currently no legislation actually out there for the use of bacteriophage in place of antibiotics. There's only been experiments and research done. So currently, actually at Purdue University, there's research that has happened through Dr. Ebner. He's done research on bacteriophage through chickens and the impact of bacteriophage to get rid of salmonella if it can. I believe his research has completed, but he is in the Department of Animal Sciences, actually. Purdue also has classes through the ABE program, ABE 226 and 227, where you actually participate in a national database and do real research to find new phages and new ways to use them for phage therapy. So ABE 226 and 227 at Purdue University are actually part of a national initiative to get more people researching and invested in bacteriophage, to find these alternatives and to find more bacteriophage to hopefully discover what all they can be used for. This is just one application, but they can be used for so much more. I've actually taken these classes, I've taken both of them, and they show just how easy it is and how invested many people are into the discovery of these uses. So as you can see, there is a website up there that is actually a database, the actual database for all the phages that have been discovered through universities that participate and also research institutes. When I took the course, I had a phage that was discovered called Maybe 2021, and as you can see in the purple there, we looked through the genome to see what the possible genes actually did, what proteins they encoded for, and then at the bottom picture you can see we actually discovered the function. And so just through this class, I participated in an undergraduate research symposium and actually got a reward because my group and I actually discovered that the gene that we looked at, the particular gene, was able to be used and had the ability to shut down some cancer-producing cells. So that may not be in the animal industry, however, it has a major impact and can be used in the future. So bacteriophage are very important. They can be used in the veterinary medicine industry because that's just their basic use. We know for sure that they can kill bacteria, and right now there's many bacteria that are becoming resistant. It's becoming harder and harder to fight off these infections. So bacteriophage are very important, and they need to be pushed through and researched so we can help our animals get rid of diseases and infections that would otherwise harm them and save ourselves money. So thank you for listening, and I hope this was informative.